Hi everyone. In the immunology subject, we are going to discuss about another topic called cells of the immune system. There are several types of cells which are involved in the immunity process of a host. So those are going to be of lymphocytes, monocytes, macrophages, neutrophils, eosinophils, null cells, APC means antigen presenting cells, and then basophils and mast cells, etc. And these are all going to be the immune cells which fight against the different types of infectious agents like microorganisms which are going to enter into the host or human body. So basically the stem cells are going to be developed into three lines. So our stem cells are going to have the lineage of three lines. The first line is going to be the lymphoidal progenitor and or lymphoidal lineage, and then the second type is myeloidal lineage, and the third one is the erythroid lineage. So that's how we are having the three types of the lineages by which these stems are getting modified into different types of immune cells. In this part, we are majorly covering the lymphoidal lineage of that means lymphoidal cells, and then in another part we will go into the detail of myeloid progenitor. And here the erythroid progenitor we are not going to touch because we are discussing about the cells which are involved in the immunity, that means the cells of the immune system. So these are the two major lineages from which we are getting the immune cells. So let's see the first one, lymphoidal progenitor or lymphoidal lineage. This is the one in which the stem cells develop into T-lymphoidal progenitor, which is going to give rise to T lymphocytes, and B lymphoidal progenitor, which is going to give rise to the B lymphocytes and null cells. And these null cells are again divided into two more types, that is natural killer cells and killer cells. Then coming to the myeloid progenitor, in which the stem cells develop into myeloid progenitor, is going to develop into some sort of a cells called as monocytes, which further get modified into the macrophages when enters into the tissue. That we will discuss in detail. Okay, and here are the rest of the cells which we call them as polymorphonucleosides. Okay, so eosinophils, basophils, neutrophils, all this comes under this one. Then coming to the erythroid progenitor, the name itself is saying that directly from the stem cells you are going to have the development of RBC cells or erythrocytes. So this is the overall division of the cells that are coming from the stem cells. That means by the three lanes, that is lymphoidal lineage, then myeloid lineage, then erythroid lineage. So here the lymphoidal and the myeloid lineage, you are going to get the cells involved in the immune system. So let's start with the lymphoidal progenitor, which is going to give rise to the lymphocytes. So what are lymphocytes? And then what are the different types of the lymphocytes? What is the difference between these lymphocytes and their functions? Everything we will discuss now. Okay, so let's see the first one, lymphocytes. Lymphocytes are going to be the central cells of any immune system responsible for acquired immunity. So this immunity types also we have discussed in another part. You can go through them. So immunity, two types, innate immunity and acquired immunity. So whatever the immunity that you are going to get because of the cells of immune system, we consider it as acquired immunity. And this is going to be of, that means the lymphocytes are going to be of about 20 to 40% of total lymphocytes present in the blood, and 99% will be in the lymph. So we had gone through the lymphoid organs also, which was synthesizing these lymphocytes, isn't it? The thymus, bone marrow. So we had gone all those things, okay, in different parts. So here, out of the total lymphocytes, 99% will be only in the lymph. So majorly that's the reason why we call them as lymphocytes, and only very few are going to be present in the blood. Then coming to the lymphocytes are going to continuously circulate in the blood and lymph and are capable of migrating into the tissue spaces. and lymphoidal organs, thereby providing a high degree of immune responses. So these are going to be like soldiers which are continuously going to have the round, that means circulation, monitoring that, such that no antigen is going to enter into the body, and that's how they are protecting our organs, tissue spaces, all the things. Then these lymphocytes are one of the classes of WBC also. Some will have the classification like that. And the lymphocyte production, differentiation and maturation takes place in primary lymphoid organs. We had gone through these primary lymphoid organs, secondary lymphoid organs, their functions, different organs, all the things in different parts. You can go through them. So here the synthesis, that is production, differentiation and maturation of lymphocytes occurs in the primary lymphoid organs, and then these lymphocytes will enter into the blood circulation and the lymph circulation and reaches the secondary lymphoid organs and tissues, where they are going to have the exposure to the antigens, and that's how they are going to have the reaction with them, that is participating in the antigen antibody reaction and removing the antigen from our body. So that's nothing but the immunity. And thus they are found in the majorly in the peripheral blood, then lymph nodes and lymphoid organs. We know that these are all lymph nodes are going to filtering of lymph, okay, and then spleen is going to filter of blood. So the mature lymphocytes or lymphocytes or lymph cells are also going to be called as navy cells. Which cells? Navy cells. And these lymphocytes which are mature, immunogenic or non-functional, as I said, they are in the primary lymphoid organs, where as they are mature, immunogenic and functional only in the secondary lymphoid organs due to the exposure to the antigen. So in the secondary lymphoid organs only they are getting exposed to the antigen. So this is all about the lymphocytes overall notes. Then coming to the functions of it, that means whatever the points regarding the lymphocytes are, these are the central cells of immune system. There are about 20 to 40% of lymphocytes present in the blood, and 99% of lymphocytes are present only in the lymph, and lymphocytes continuously circulate in the blood and lymph and are capable of migrating to the tissue spaces, lymphoidal organs, and thereby providing a high degree of immune response. And lymphocytes are going to be produced, getting differentiated and matured in the primary lymphoid organs, and they enter into the blood circulation and the lymph circulation and reaches the secondary lymphoidal organs, where and become there functional because of the exposure to the antigens. Then coming to the function of these lymphocytes, obviously they will recognize of antigen lymphocytes, have antigen recognition mechanism on their surfaces, enabling each cell to recognize only one antigen. This is very important. That's why the antibodies and the antigens are going to be specific with one another. The recognition of the antigen lymphocytes have antigen recognition mechanism on their surfaces, enabling each cell to recognize only one antigen. And not only that, induction of immune response or tolerance to specific organs is also going to occur. And not only that, coming to the B lymphocyte, the storage of immunological memory also occurs. Okay, so here is about the introduction to the lymphocytes and then functions of the lymphocytes. Now, coming to the classification of the lymphocytes. The lymphocytes are again divided into three main groups based on their function and the surface markers that are present on them. So according to these surface markers and functions, the lymphocytes are going to be classified into three types. How many types? Three types. The first one is going to be the T lymphocytes, the second one is going to be the B lymphocytes, and the third one is going to be the null cells. T lymphocytes, B lymphocytes, and then null cells. So here we are going to discuss in detail about the T cells or T lymphocytes, then B lymphocytes or B cells, and even the null cells. So let's go with the first type of the lymphocytes, that is T lymphocytes. The pro T cells or precursor of T cells, which often called as prothymocytes, are produced in the bone marrow. Yes, we had discussed this in the primary lymphoid organs. Each and every cell of a body is going to be synthesized in the bone marrow region only. And these T lymphocytes are also going to be produced in the bone marrow, but they are going to be called as prothymocytes. And these prothymocytes enters into or migrates to the thymus organ and undergo development, maturation and differentiation. So these processed cells are now called as T lymphocytes or thymus derived cells. Remember, the cells that were produced in the bone marrow are going to be called as prothymocytes, and we are not going to call them as T lymphocytes or T cells because they don't have any hormone stimulation or any surface markers. They are undifferentiated. But these prothymocytes, once enter into the thymus organ, they undergo a differentiation, that means development occurs, then maturation occurs, then differentiation also occurs. And now these differentiated, developed, matured lymphocytes are going to be called as T lymphocytes or T cell or thymus derived cells. Okay. Now some T cells are transported away from the thymus and enters the bloodstream, where they comprise about 70 to 80% of the circulating lymphocytes. Other T cells tend to reside in various organs of the lymphatic system, such as lymph nodes and the spleen. So that means, for example, 100% of the T lymphocytes were produced, out of which 70 to 80% are going to have a duty in the blood circulation, so that they can monitor all the things throughout what we call it as monitoring the things through the circulation. Okay, and next to the 20%, like a patrolling. Okay, the police will do the patrolling, no? So in the same manner, here you are going to have the 70 to 80% is going to have the patrolling work in through the bloodstream, and the rest of the 20% lymphocytes are going to be present in various lymphoidal systems like lymph nodes and the spleen. Okay, so that means they reside at the stations where they have to look after. Then coming to the these T cells produce some sort of lymphokinin substances, and they are mainly responsible for cell-mediated immunity. But it can also help humoral response. Remember, the T lymphocytes are mainly responsible for cell-mediated immunity, but it can also help the humoral response. How we will discuss in detail now. Based on the surface markers, MHC restriction, that is most histocompatible restriction, target cells. Then coming to the target cells and functions, T cells can be again classified into two types. Okay, so based these T lymphocytes are again classified into two types. So basically lymphocytes are classified into three types, isn't it? T lymphocytes, B lymphocytes and null cells. But depending upon the what we call it as surface marker, then MHC restriction, target cells and functions of T cells, these T lymphocytes are again classified into two types broadly. That is, one is called as TR and another one is going to be called as TE. So what is a TR? The TR is the T regulator cells and TE is the T effector cells or effector T cells. And these are further classified into two to each type, that is T regulator cells are again of two types, that is T helper cells, T suppressor cells. In the same manner, T effector cell or effector T cells are again of two types, that is T cytotoxic cells and another one is a T delayed type hypersensitivity cells. That means the T cells which are involved in the delayed type of hypersensitivity are going to be called as TDTH. Okay, so that's how the T lymphocytes are broadly classified into two categories. One is the T regulator cells and second one is the T effector cells or effector T cells. Okay, let's see them in detail now. The T regulator cells develop into TH or TS. That's what I told you, T helper cells, T suppressor cells. and control the balance of between the enhancement and the suppression of antibody formation. So this is how they are going to the T lymphocytes. Here I told you a point that it can also help in the humoral response. So these T regulator cells are going to be involved in the humoral response. Whenever we are going to have a need of having more production of antibodies, these T helper cells are going to activate the B cells such that they are going to produce the antibodies. So if the antibody production do not require, then these T suppressor cells will suppress the production of antibody by telling it. So this is how it is going to regulate it, what that it is the production of antibody. So these cells develop into TH or TS cells and control the balance between the enhancement and suppression of antibody formation. So let's see the first one, T helper cells. These cells are necessary for B cell activation by T dependent antigens and for activation of T effector cells. Then coming to the T suppressor cells, these cells suppresses the activation of T helper cells and B cells. Okay, so that's how the name itself is saying it is going to regulate it, whether the antibody production or formation is required or it have to be suppressed. Okay, then the second type of the uh, T lymphocyte is effector T cells, simply denoted as TE, and these are again of two types. One is the T cytotoxic, another one is the T delayed type hypersensitivity. The T cytotoxic cells, these cells bring about cytolysis and death of the cells recognized as non-self, that is antigens, and parasitic infected cells. So they directly target the non-self cells recognized and the parasitic infected cells, and they will lyse them. So that's how the T cells involved in the lysis of the cell is going to be considered as T cytotoxic cells. Then coming to this another type of T effector cells, that is T delayed type hypersensitivity, that is TDTH. These cells provide protection against intracellular infectious agents and mediate inflammation and activate macrophages in delayed type of type 4 hypersensitivity. We will have this discussion of hypersensitivity in detail in another type of another video or another part. You can go through it. So here this TDH type of the cells are mainly involved in the delayed type 4 hypersensitivity. And now the T cells are classified into different antigenic subsets based on their surface markers or surface glycoproteins and designed with a name called as CD number, that is cluster differentiation number. The CD refers to the cluster differentiation. So you may heard about a CD8, CD3, CD4 cells. So these are also the types of T cells only. So these T cells, if you are going to have some sort of a surface glycoproteins and designed with some sort of a number called as CD number, and that CD is nothing but the cluster of differentiation, a designation assigned to leukocyte cell surface glycoprotein molecules. As I told you, we had different types of identified this CD cells, that is CD3, CD4 and CD8 cells, and this CD number is going to be mainly going to be useful in some of the diagnosing some diseases or infections, especially in the HIV identification. Okay, then this is all about the T lymphocytes that we have discussed. That is, there are two types mainly, regulator T cells and the effector T cells. And then the regulator T cells, that is TR cells, are again of two types depending upon the suppression or enhancement of B cells production, that is antibody production, that is TH and T suppressor. And then coming to the effector T cells, again we had two types, that is TC, that is cytotoxic T cells, and TDH, that is delayed type hypersensitivity T cells. So this is all about the T lymphocytes. Then moving to the B lymphocytes, the second lineage or second type of lymphocytes is B lymphocytes. So here obviously we know that B lymphocytes are derived from bone marrow. The T lymphocytes are also derived from the bone marrow, but we call them as prothymocytes. Okay, so here the lymphocytes derived from bone marrow are called as B lymphocytes or B cells. I told you the bursa fabricus is an organ especially present in the birds. So the bursa fabricus is going to produce the B cells but not the bone marrow. Now, the distribution of B cells is high in the spleen, lymph nodes, and bone marrow and other lymphoidal tissue, but low in the blood. Whereas the T lymphocytes are more in the number, that means 70 to 80% will be present in the blood, and only the rest of the 30 to 20% will be going to reside in the tissues and organs of the lymphatic system and other. Whereas here it is different. That is, it is quite opposite. The B cells are going to be mainly present in the organs, and in the circulation we will see only very less number of B cells. And there are only about 20 to 30% in the circulating lymphocytes. Okay. These circulating B cells contain a surface immunoglobulins, so such as IgM and IgT. So majorly in the circulation of these type of B cells, you are going to see these two type of immunoglobulins, that is IgM and IgD. So the IgM is a major receptor for its appropriate antigen. So this is the reason why the recent infection is going to be identified by the presence of IgM majorly, because this is the one which immediately reacts with the antigen. Okay, then moving to the next, these B cells are also having some sort of receptors on their surface as FC, that is fragment of crystallization part of some immunoglobulin classes, and may have also some other receptor components like C3b. Okay, this C3b is a complement, so complement system. So this also we are going to have it in another part. So all the B cells are going to possess two receptors. One is the FC part, another one is the complementary part, that is C3b. Each B cell may have as many as 10,000 of these specific antigen receptor cells. So that's why it is very active and going to be participate more than that T lymphocytes. And coming to the primary development of B cells is going to be under the genetic control. That we know, the every cell is going to be synthesized under the genetic control. So here the primary development of B cells are also going to occur under the genetic control and takes place in the bone marrow throughout the life. Okay, maybe in a low level. The secondary development of B cells occur in the germinal center of secondary follicle. So in the secondary lymphoid organs, we have seen this primary follicle, secondary follicle, all these things. And there you can have the clarification of what all these secondary follicles are, isn't it? And these secondary follicles are going to give the development of B cells, and thus leading to the formation of plasma cells, which further gives rise to the antibodies. So some activated B cells becomes a long-lived memory cells also. So we are going to discuss about what are the different types of B lymphocytes now. Okay, so for the development of this secondary development of B cells, it requires the active cooperation of the T helper cells. So that's how the T helper cells are going to have the involvement in the regulation of what we call it either suppression or enhancing the B cell production. So T helper cells are required for the secondary development of these B cells. Okay, and B cells produce antibodies, so that we know very well that B cells are the one which is producing the antibodies. Now these B cells or B lymphocytes are going to participate in humoral mediated immunity, whereas the T cells or T lymphocytes are going to participate in cell mediated immunity. Okay, so now these B lymphocytes or B cells are again of two types. One is going to be called as a memory cell, another one is going to be called as plasma cell or effector B cells. Let's see what is a plasma cell, and then we'll move to the memory cell. The plasma cells, they do not have any surface markers and they generate a pool of antibodies. That's all. That is about 2000 per second during their lifespan. That means one plasma cell will give these many antibodies. And these are short-lived and having a lifespan of only 5 to 7 days. And these are going to be called as N cells. Whereas the memory cells, these cells are having a long life. So memory means you are going to have the long life. So these memory cells are the long-lived one, whereas the plasma cells are the short-lived one. The memory cells are going to be more very actually from blood to lymph and survive for longer time. And these respond quickly and effectively during a subsequent similar antigenic stimulation occurs. So that means if you are having this second exposure to the same pathogen or antigen, then these are the cells which are going to be involved. Okay, so this is a area of the cells where we are going to have the vaccination mechanism working on it. Okay, so here the B lymphocytes, as I told you, you are going to have the two types, that is the plasma cells and the memory cells. And from the plasma cells, you are going to have the production of antibodies. And each plasma cell can give rise to of nearly 2000 per second antibodies. And these are having very short life, that is only from 5 to 7 days, whereas the memory cells are going to have a long life, long-lived one, and these are mainly involved in the secondary immune response. Okay, so this is the structure of a lymphocyte that you can go through. And the third type of the lymphocyte that we are going to discuss is a null cells. So a small portion of lymphocytes circulating in the blood, that is of about 5%, are not having any type of surface markers or surface receptors like the T or the B cells. And they are failing to synthesize the antibodies, and those are being called as null cells. Null means nothing. So they are not going to have any surface markers on them, and those are being named as null cells. And these null cells are going to be large with abundant cytoplasm containing granules called as eosinophilic, and hence these are also known as large granular cells or lymphocytes. Large granular cells or large granular lymphocytes. And these null cells are again of two types. One is natural killer cells, another one is the killer cells. Coming to the natural killer cells, there are large granular cells. So as I told you, these null cells are going to be of large in size. So here natural killer cells are also going to be of large granular cells. And these are important to fight against the virus infections and malignant cells. Okay, so this is what you have to remember here. The natural killer cells are mainly important to fight against the virus infections and the malignant cells, and they directly kill the malignant cells. Malignant cells means nothing but the cells which are causing the cancers, and even the virus infected cells by non-specific mechanism. So here there is no specificity. So just if they are going to identify any virus infected cell or any malignant cell, they will target them and they will demolish it. So they play an important role in severe combined immunodeficiency diseases. So these are going to be very crucial in severe combined immunodeficiency diseases where both B and T cells are going to be absent. At that, the cells which are protecting those immunocompromised persons are nothing but these natural killer cells. And these natural killer cells can be activated by interferons and interleukin cells. Okay, and these are antibody independent cells. The natural killer cells are not bothered about the antibody cells and show some type of a natural system to kill the antigen. Like that, how the macrophages and other leukocytes are going to kill them. In the same manner, the natural killer cells also kill the virus infected cells or the malignant cells directly without depending on the antibodies. So this is all about the natural killer cells. Then coming to the killer cells, these cells possess some sort of surface receptors like Fc portion of IgG antibody. So IgG antibody possesses two parts, isn't it? FAB part and Fc part. So IgG antibody Fc part is going to be having on these cells. So and those cells are going to be called as killer cells. They are capable of killing or lysing the target cells sensitized with IgG antibody. So these killer cells are the dependent on antibodies because the antigens which are coated by this IgG antibody are going to be recognized by these cells and they are going to target. And that's how we call these killer cells as antibody dependent cellular cytotoxicity, ADCC, antibody dependent cellular cytotoxicity, cells or killer cells, killing. So these cells are inactive in the absence of specific antibody. So, but that is not the case with a natural killer. That means they are directly killing, no need of any help. By birth, they have come like that. Okay. So now, but the killer cells have to be take the help of this IgG antibody, and then only it can do it. And these are non-phagocytic killer cells, gives protection to the organism by destroying the pathogens like bacteria, virus, fungi, etc. Okay, so this is all about the killer cells. So that's how we have discussed all the three types of lymphocytes, that is, one is the T lymphocyte, then we had gone with the B lymphocytes, then the null cells. So T lymphocytes are of two types, regulatory and effector, which are further classified into helper cells, suppressor, cytotoxic and delayed type hypersensitivity. And the B lymphocytes are classified into two types, plasma cells and the memory cells. And from the plasma cells, you are going to have the production of antibodies. And coming to the null cells, where there will be no surface markers related to the T or B, those are called as null cells. And these are again of two types, natural killer cells and killer cells. Natural killers are independent of antibodies reaction, whereas the killer cells need the help of the antibodies. That's why we call them as ADCC. Okay, antibody dependent cytotoxic cellular cytotoxicity. Okay, so this is all about the lymphocytes. And then we will discuss the myeloid lineage, that's the other cells which are involved in the immune system or cells of the immune system in another part. Thank you.